So, my dear zoology students, in continuation of the online class, today let us discuss about the glycolysis. Glycolysis, this is a portion of the biochemistry portions. Now let us start about the glycolysis. Glycolysis is the conversion of glucose or glycogen into pyruvate and lactate. Lactate is produced from pyruvate only when oxygen supply to the tissue is insufficient. Under aerobic conditions, pyruvate enters the TCA cycle through acetyl-CoA for the further oxidation of glucose. All the enzymes involved in glycolytic pathway are found in extramitochondrial solvent fraction of the cell. Glycolysis takes place in the cytosol of a cell, and it can be broken down into two main phases: energy requiring phase and energy releasing phase. Each reaction in glycolysis is catalyzed by its own enzyme. Now first let us discuss energy requiring phase. In this phase, 2 ATP are spent to form an unstable sugar with 2 phosphate group, which then split to form 2 3-carbon molecules that are isomer of each other. Each step is catalyzed by its own enzymes. Energy requiring phase consists of 5 different reactions. In first reaction, glucose is phosphorylated to glucose 6-phosphate by ATP in the presence of hexokinase and the magnesium ions. It is an irreversible reaction in most tissue. However, liver and kidney tissue contain a specific enzyme called glucose 6-phosphatase for the reversal of this reaction. In the second step, glucose 6-phosphate is converted into its isomer fructose 6-phosphate by the enzyme phosphohexose isomerase. In the third step, fructose 6-phosphate is phosphorylated to fructose 1,6 biphosphate. This step is catalyzed by the enzyme phosphofructokinase, which can be regulated to speed up or slow down the glycolytic pathway. It is an irreversible reaction in most tissue excepting liver and kidney. In the fourth step, fructose 1,6 biphosphate splits to form 2 3-carbon sugar, that is dihydroxyethanol phosphate and glycerol dry 3-phosphate. By the enzyme aldolase, they are the isomer of each other, but only one glycerol dry 3-phosphate can directly continue to the next step of glycolysis. In the last step of the energy requiring phase is the dihydroxyacetone phosphate is converted into glycerol dry 3-phosphate by the enzyme phosphotriose isomerase. Then come to the energy releasing phase. Energy in the second half of the glycolysis, the 3-carbon sugar formed in the first half of the process, that means in the energy requiring phase, go through a series of addition transformation, ultimately turning into pyruvate. In the process, 4 ATP molecules are produced along with 2 molecules of NADH. The different reactions in the energy requiring releasing phase are shown here. In the first step of the energy releasing phase or 6th step of the glycolysis, glycerol dry 3-phosphate is oxidized to 1,3-biphosphoglycerate. This reaction is catalyzed by the enzyme glycerol dry 3-phosphate dehydrogenase in the presence of inorganic phosphate and the coenzyme NAD+, which is reduced to NADH and the hydrogen ion. Under aerobic condition, the reduced NAD is oxidized through electron transport chain of mitochondria, resulting in the formation of 3 ATP molecules. So, in this step, 6 ATP molecules are synthesized. In anaerobic glycolysis, no ATP is formed as the NADH is deoxidized during the conversion of pyruvate to lactate. In the next step of the energy releasing phase, 1,3-biphosphoglycerate donate one of its phosphate group to ATP, adenosine diphosphate, making a molecule of ATP and turning into 3-phosphoglycerate. In the process, this reaction is catalyzed by phosphoglycerate kinase in the presence of magnesium ions. Thus, at this stage, 2 ATP molecules are formed per molecule of glucose. And then in the next step, 3-phosphoglycerate converted into its isomer 2-phosphoglycerate by the enzyme phosphoglycerate mutase. Then in the next step, 2-phosphoglycerate loses a molecule of water, becoming the phosphoenol pyruvate. This reaction is catalyzed by the enzyme enolase in the presence of magnesium ions. In the last step of the energy releasing phase or the last step of the glycolysis, phosphoenol pyruvate readily donate its phosphate group to ADP, making a second molecule of second molecule of ATP. As it loses its phosphate, PEP is converted into pyruvate, the end product of the glycolysis. This reaction is catalyzed by the enzyme pyruvate kinase. This is about the process of the glycolysis. Then next, let us discuss about the energetic of the glycolysis. This is the energetic of the glycolysis. Number of ATP formed during the oxidation of one molecule of glucose in aerobic glycolysis. In the step, in the conversion of glycerol triphosphate to 1,3-diphosphoglycerate, 6 ATP molecules are synthesized. In the conversion of 1,3-diphosphoglycerate to 3-phosphoglycerate, 2 ATP are produced. In the conversion of phosphoenol pyruvate to pyruvate, 2 ATP molecules are produced. So total number of ATP produced is 10. And in the conversion of hexokinase and the phosphokinase reaction, 2 ATP are consumed. So net ATP formed is I in the case of aerobic glycolysis. In anaerobic glycolysis, reduced NAD in glycerol diphosphate dehydrogenase step is not reoxidized in the mitochondria, and therefore net ATP formed will be only 2. This is about the glycolysis. Thank you for watching, my dear students.